Next is about garbhavyakarana shariram vyakhyasyaamah yathovatadbhuvantanantarihi. Garbhavyakarana is Vyakarana. The word means analysis. Analysis of the fetus and the fetal body is the issue. Now, Agni Somo Vayu Sattvam Rajas Samah Panchendriyani Bhutatma iti Pranaha Tasya Khalu Evam Pravrittasya Shukrashuntasya Abhipatyamanasya Kshirasyeva Santanikaha Saptatvacho Bhavanti. तासां प्रथमा अवभासिनी नाम या सर्वान् वर्णान् अवभासयति पञ्चविधां च छायां प्रकाशयति सा व्रीहेः 18 भागप्रमाण सिद्धपद्मकण्टकाधिस्थाना द्वितीया लोहिता नाम 16 भागप्रमाण तिलकारकन्यच्च व्यङ्गाधिष्ठाना तृतीया श्वेता नाम 12 भागप्रमाण चर्मदर अजगल्ली मशक अधिष्ठाना चतुर्थी ताम्रा नाम 8 भागप्रमाण विविध किलासकुष्ठाधिष्ठान पंचमी वेदिनी नाम 5 भागप्रमाण कुष्ठविसर्पाधिष्ठान षष्ठी रोहिणी नाम व्रीहिप्रमाण ग्रंथि अपचि अर्बुद श्रीपद गलगंडाधिष्ठान सप्तमी मांसधरा नाम व्रीहिद्वयप्रमाण भगंधर विद्युदि अर्षो अधिष्ठान यदेतत् प्रमाणं निर्दिष्टं तन्मांसतलेषु अवकाशेषु नललाटे सूक्ष्माङ्गुल्यादिषु च यतो वक्ष्यति बुधरेषु व्रीहिमुखेन अंगुष्ठ उदरप्रमाणम् अवगाढं विद्येते इति. Now the formation of the body after the fertilization, once the zygote is formed, and the next structure, the levels of the formation of the body. Of course, now we have referred to that issue, like the formation of the gastrula, morula, and then the 3 germinal layers, and then from the germinal layers you will have the different structures. Out of them, the ectoderm would result in the formation of the outer coverage of the body, the skin. So Sushruta starts with the outer covering structure of the body as the tvak. So to translate tvak as skin, again there is some reservation, so I will come to that part. So Sushruta refers to the formation of the tvak as the one example of the structure. It's not that the tvak is formed in the beginning, so it should not be considered that way. Instead, from the assessing of the fetal body, the prominent structure, the most or the rather biggest of the organ of the body is the skin, the outer surface, and hence Sushruta has considered the tvak as the first of the structure to be discussed. Not that the skin is formed in the beginning. So some of the commentators say that the skin is formed in the beginning. That's not true. Sushruta never says that. Instead, it is said like the formation of the structures will go or form in a gradual process, and that process of the formation of the structures of the body are compared to the formation of the cream in the milk. When you boil the milk, the cream is formed. The cream is formed as a gradual process, and the layers of the skin, this cream, would be formed one by one, and gradually there will be condensation. Similarly, the structures of the body also are formed, and even the tvak also is formed in the same manner. And according to Sushruta, tvak has 7 layers, and those are avabhasini, the first layer. The word avabhasini means which can show you the appearance, gives the appearance, avabhasa. It gives you the appearance as such. And because it sarvan varnan avabhasayati, it makes the color of the body, hue of the body visible. That's why it's called as avabhasini. And panchavidham cha chayam prakashayati, it gives that the different varieties of the chaya, the hues of the body. Now, hues of the body, we usually use the term fair, black, and so on, dark, and so on. Sushruta says there are 5 varieties, degrees of that fairness and degrees of the darkness, and based upon that 5 varieties are there. And according to Sushruta, the thickness of that would be 1/18th of the vrihi. One vrihi is around 1 centimeter, roughly. Not that it's exactly 1 centimeter. Vrihi dhanya, the length of the vrihi dhanya is the supposed dimension of the thickness of the tvak in the thickest part of the body. Now, thickness of the skin is not uniform throughout. Sushruta says like lalate sukshmangulyadishu, in the over the forehead or in the other fingers like area, the thickness is lesser, Whereas in the thicker part of the body, like the muscular portions, the flabby portions of the body, like the abdominal wall, buttocks, the thickness is mentioned. Whatever the measure of the thickness mentioned here is related to such areas of the body where the muscle, this body is flabby, or like abdominal layer or the buttocks as such. And there the thickness of the first layer is Ashtadasha Bhaga Pramana, 1/18th of the is Brihidhanya. And Sigma Padma Kantakadisthana, the location, the diseases like Sigma, that's the discoloration, hypopigmentation, and the keratinization, Padma Kantaka, hyperkeratinization, roughness, skin being rough. Such lesions are located over the avabhasini layer. Now, if you see the microscopic anatomy of the skin, generally when we study the microscopic structure of the skin, we refer to the skin into two layers, epidermis and dermis. But if you go into further microscopic structure of the epidermis, the layers of the epidermis, the first of them would be the keratinase layer. Now, the formation of the skin has a very classical manner. The skin cells, they divide at the basal layer, and then after that there is a gradual decomposition, like that word decomposition is not a very correct word. There is a gradual formation of this skin, the cells, into a non-living substance, and that outermost surface of the skin is a non-living substance, is called as keratin. Keratin is a word suggests about a complex molecular structures which have fatty contents and which are partially water soluble. This is partially water soluble, and it's scalable. It can be scaled as such. And because of this kind of a situation, where the skin gradually, the tissue, a living tissue, gradually desiccates, shrinks, and then converts into a non-living substance. And that non-living substance also is a part of the skin, which we call as the stratum corneum, the keratinase layer. That's the most superficial layer. During this formation of this keratin, the cells will show some changes. Gradually, the cells, initially the basal cell layer, which has all the living characters, the cells are usually having this hexagonal shape. Nucleus is quite big, and the cytoplasm also is quite huge. Gradually that gets reduced, the cytoplasm gets reduced, the nucleus becomes smaller and smaller, and hence those cells, they look like pointed spines, like, and that's why that layer is called as stratum spinosum. And then as it further goes down, like the deterioration occurs, it results in a situation where the cytoplasm becomes completely reduced or significantly reduced, and all that you see is some granular layer with some very small nuclei. That's a stratum granulosum, and then it becomes a situation where it becomes somewhat transparent. The structure becomes somewhat transparent, and relatively thin consistency comparatively, not as thick as the cells. That's the stratum lucidum. Then it dries up, and then it results in keratin layer. Now that keratin layer doesn't have the pigmentation, but because it is semi-transparent, it allows the color of the melanocytes to be seen over the surface, and stratum granulosum and stratum lucidum as well as stratum corneum, they are somewhat semi-transparent, and hence the skin, you will be able to see the colors over there, and hence Savaranam Abhasayati. So Varana, the color can be seen through the keratin tissue layer. And whatever we say as the dark hue or the white hue is mainly dependent upon the keratin tissue layer. You have plenty of diseases where the diseases of the keratin tissue synthesis would affect, like psoriasis, where there is a hyperkeratinization, or atopic dermatitis, where the keratin tissue will be deficient, and hence the deeper layers of the skin are exposed as such. Now that's exactly Sigma Padma Kantakadisthana. So it's exact description of the zona keratinosa or keratin layer, the avabhasini. The next is Dvitiya Lohita Nama, Shodasha Bhaga Pramana, Tila Kalaka Nyacha Vengahadisthana. The next is the Lohita. The color is reddish in color. The Lohita suggests about the reddish in color, and it's about 1/16th of the thickness of the Vrihi, And the diseases of pigmentation, hyperpigmentation, like moles, they are located in these areas. That's what Sushruta says. But as we know, the moles now are considered to be located at the basal layer, but they will be but the melanocytes are located at the basal layer, and gradually, actually, as they get deteriorated, the lesions occur. Actually, they will involve, the dead cells will involve all the layers. Now, anyway, whatever Sushruta has considered as the Lohita, it relates to probably the stratum granulosum and the stratum spinosum, where these cells are still living, and melanocytes also are seen at that area, and we can somewhat say that the description is resembling that of the Lohita, the second layer. Then the third layer is Shweta Nama. This third layer is Shweta, named as Shweta, because the color is somewhat paler, and it's Dvasasha Bhaga Pramana, 1/12 of the thickness of the Vrihi, and it produces Charmadala Adagalli Mashakadisthana. The diseases like Mashaka, again a pigmentation disorder, Charmadala, where there is a skin cracking, the elasticity of the skin being affected. Of course, the elasticity of the skin is dependent upon the dermis layer, the deeper layers, like as we know, the collagen tissue is more there. But if the collagen tissue is broken down, the changes are seen on the surface, and hence Charmadala may be considered as the third layer, the layer of germinal layers. These germinal layers can be considered as the Tritiya. Now, though basically the elasticity of the skin is based upon the collagen tissue layers of the deeper plane. If the collagen tissue breaks down, the immediate effect will be on the growth of the skin, and hence there will be cracks formed as such. So that's about the layer of Tritiya Shweta Nama Charmagala Jagadika. Then Chaturthi Tamra Nama Astabhaka Prabhana Vividha Kilasa Kustavistana. The fourth layer is Tambra. Tambra again suggests of red color, and the Lohita also is red color, but Tambra is much redder, bright red than the Lohita, and that suggests about the vascular entity, the entity where there will be more of the blood flow. Now, if you look at the dermis, below the epidermis, now whatever we have discussed till now is the epidermis. The dermis layer, in the classical anatomy, we differentiate them into two categories, the papillary and the reticular layer. The papillary layer is the layer where you will have plenty of blood vessels and the nerve endings. Nerve endings also are present over there. The sensory area would be here, and if you cut the skin up to the layer of the dermis till the layer of germinal layers, there will be no bleeding, no frank bleeding. There could be some serous discharge. But when you cut it below the dermis, there will be bleeding. Now that's exactly the fourth layer of the Tvak, as mentioned by Sushruta. Then Panchami Vedini Nama, the fifth layer is considered as Vedini. Now that because it gives the sensation of the body. And the lesions like Kushta, Visarpa, etc. are located at that place. Now these Vedini and the Tamra, these suggest about the papillary layer of the dermis, where you have the vascular and the neurological bundles together, and blood supply of the skin as well as the sensory areas. They are present in these zones as such, and hence we can consider the Tamra and the Vedini as the papillary layer of the dermis plane. Then in the reticular layer of the dermis, the tissue is mainly the elastic collagen tissue which gives the elasticity to the skin and the stress tolerance of the skin. The strength of the skin is based upon that area. Now that's what is considered as a Rohini by Sushruta. Rohini is the word suggests about something which allows the growth, which keeps the strength as such. Rohat, the growth is mentioned like, and that's according to Sushruta, it's a Vrihi Pramana, thickness. Of course, from the current point of view, thickness of these skin areas, zones of the skin can vary from area to area, as Sushruta has already mentioned that they can vary from area to area. as well as it can vary from species to species. In a human species also, maybe from certain types of the human being to the others, the thickness of the skin can vary. So there are definitely some people who are thick skinned and who are thin skinned in the other way also. And that's the area where, according to Sushruta, Granthi, Apachi, Arbuda, Shlipada, the diseases like nodular lesions. Among them, the most quite important is the lymphatic disorders. The maximum of the lymphatic vessels, they are present in the reticular plane. Of course, lymphatic capillaries also are present in the papillary layer also, but majority of the lymphatic vessels are presented in the reticular layer as such, and conditions like lymphedema, they would be presented more in these layers as such. The next part is the hypodermis. The other way we categorize them into the layers like epidermis and dermis and the hypodermis. Though this hypodermis is not considered as a part of the skin, there is a practice of calling this third layer as the part of the hypodermis as such, which is really the fat layer, the adipose tissue layer. And that adipose tissue layer is considered by Sushruta as the mansadhara. The mansadhara is the layer, the last layer of the tvak. And that's why I would not translate tvak as the skin. Skin and tvak are not synonyms. Because when we say the skin is related to the dermis and epidermis. The hypodermis is not part of the skin. Though we use the word dermis there, it's not a part of the skin from the current anatomy point of view. But from Sushruta's point of view, tvak includes that layer too, because it also has a resemblance to the functions of the skin in the way, like it can cover the area of the body. Of course, the mansadhara kala may have some more functions other than that of the issue like the tvak as such. There are other layers of this tvak. And the deep-seated diseases like Bhagandara, Vidradi, Arsha, the diseases like septic pathologies, abscesses, they are located at this plane according to Sushruta. That's how the layers of the tvak are seen, and they are exactly described in a almost similar fashion. But you get now more details. You can have more objective assessment of these layers under the microscope. But these layers are described by Sushruta as the layers of the tvak. Then the next again would be a bit of controversial issue is kalaha. The word kala means the exact Sanskrit literature. The word kala means a segment, a part. Now, but in Sushruta, the word kala has a separate meaning. It's a membranous structure which gives you the identity of different structures of the body, or something like the layers in between different organs or different planes of the tissue, which we now call as the planes of the tissue. Kalaha khalvapi sapta bhavanti dhatu ashayantara maryadaha. The kalas are the layer-like structures. I would not say the word layer, but the specific differentiating structures between any of the dhatus or ashayas, any of the body cavities, visceral organs of the body, or it could be in between the two planes of any of the tissues of the body. Or if there is a confusion, she should give you some more clarification. Clarification of the kala. Yatha hi sarakkasyeshu chidyamaneshu drishyate tadha hi dhatuhu mamseshu chidyamaneshu drishyate snayudhishta pradichannan santadamsya jarayuna shleshmana vestitamsyapi kala bhagamstutam viduhu. Like if you cut a wood, now you will see the layers of the different, you will see the bark of the wood, then the pith of the wood. In between you may see some circular structures, rings in the wood, which suggests about the age of the wood. Now these separating structures, the structures which are visible and which can be, which can actually differentiate it into two different or maybe multiple different planes like the pith, the bark or the so on. Similarly, when you cut the tissues of the body, you will be able to see such structures, and such structures which are covered by certain either a express cover layer, which we use the term fascia, or at times it may not be directly visible, the these visible or expressless face structure, but it could be like there is a differentiating margin, and that's how the color are to be understood. Color are to be understood as a either the fascias, or they could be considered as a the margins of different layers of the tissue, which help in differentiating the structures as it. Among them, Tasam Prathama Mamsadha Nama Yasyam Mamse Sirasnai Dhamane Srotsam Pratanaha Bhavati Yatha Bisamradani Vivardhante Samantaraha Bhumo Pankodakastani Tata Mamse Siradeha. Now, the Mamsadhara layer of the Tvaka and the Mamsadhara Kala, which helps in differentiating the structures, they are probably the same. The difference is that when it is a part of the Tvaka, it's considered as a Mamsadhara layer of the Tvaka. When it has to be considered as the total body structures and the differentiating planes, then it has to be considered as a Mamsadhara Kala. But when we say Kala again, there has to be some more differentiation. That differentiation is a differentiating between the structures of the muscles and the skin, in between the interface of the muscles and the skin. They are to be differentiated, and that differentiation is done by a layer of the adipose tissue, and that layer of the adipose tissue again has a clear cut two differentiating planes, which we call as a campus fascia as well as a scarpus fascia. The campus fascia is just below the skin, and a thin layer which can be differentiated or dissected with difficulty. That dissection in surgical practice, when you make an incision, this is one of the important zones of that making of the incision. And if you cut the skin and you have to differentiate, this dissection is not simple, it's not easy. You will have to go for gradual dissection, maybe a blunt dissection, to separate that. Whereas if you go deeper into the layer, and then it has to be differentiated from the muscles, the scarpus fascia, it's thicker, well developed, and it can be easily differentiated or easily dissected out. So when you look at the skin layers of the body, abdominal layer maybe, the deeper layers of the muscles, but above the muscles you will have a thick plane. That's what we call as the scarpus fascia. Scarpus and campus fascia have a lot of significance in the diseases like hernia, in the hernial sacs, differentiation of the hernial sac and hernial surgery. These fascias have to be differentiated clearly, and they need to be dissected out. And during surgical dissection of any wound, any surgical incision, these layers have a significance to play. And the adipose tissue in between is the area where you will have plenty of blood supply. The blood vessels are there, and this blood supply would be reaching to the tissues. So fat tissue or the adipose tissue there has many other functions. So that Mamsadhara Kala may be suggestive of the adipose tissue, but it's not only the adipose tissue as we consider in general as the fat tissue. So often adipose tissue is translated as fat tissue and gives an impression as if adipose tissue is only responsible for the fatness. But that's not true. Adipose tissue is not the only responsible for the fat tissue. Rather, whatever we call as obesity is more dependent on the storage of water than really the fat. Of course, fat also is true, but more than the fat, it is the water which is more significant. And the adipose tissue also would have different types of the adipose tissue, and their functions also are different. Based upon the type of this adipose tissue, they are called as categorized as white or beige or brown. White is it's more of fat is accumulated, and the main function of this white tissue is the storage, whereas beige tissue, it has the other metabolic functions, and brown is also having the metabolic functions. The major areas of the collection of the adipose tissue is not only the subcutaneous plane. In the subcutaneous plane too, the layers where they are quite thick, they are huge, would be in the abdomen, in the gluteal region. In addition to that, the other major areas where the adipose tissue is stored is in the mesentery and the epicardial area. The functions of these white, brown and the beige adipose tissue, they are different because the functions of white is mainly storage of the energy, and the brown tissue is mainly responsible for exchange of the energy. See, whenever the body always prefers to source the energy from glucose. When there is excess of the glucose is converted into glycogen and it is stored. And major source area where the glycogen is stored is in the liver as well as in the adipose tissues of the body. And that's how it becomes a relatively white adipose tissue. Whenever there is a relatively lesser provision of the glucose and the body has to shift over to other options, there could be many means. One is the glycogen may be converted into glucose and gluconeogenesis occurs and then the metabolism would continue. Or if this is not enough, the fatty acids, the triglycerides, they themselves would come into the picture and they will be metabolized. There it will end up in a metabolism where it's not completely metabolized into carbon dioxide and water. Instead, you will have some fatty acids produced and hence it can act as a source of energy as well as it produces heat. The beige adipose tissues, they produce mainly the heat. They cover the body and preserve the heat as well as whenever there is a need for this heat energy, they can generate the heat. The locations of these white and brown adipose tissue are mainly, the white adipose tissue is mainly in the subcutaneous area throughout the body and intra-abdominal and epicardial and gonadal in the testes. Whereas brown adipose tissue more seen in interscapular, paravertebral, perineal, cervical and supraclavicular areas. And of course, the beige tissue is usually produced due to conversion of a white adipose tissue by the stimuli for production as such. Their shapes are different, microscopically they can be differentiated as such, and the composition of these cells also are different as such. The other functions of the adipose tissue, it's not the adipose tissue alone, adipose tissue has the function of only the body coverage, heat preservation, that's not the only function. It has many other metabolic functions. So adipose tissue is a metabolically active substance, and it's not that the adipose tissue is only responsible for the weight gain. It can be has many these hormones which are responsible, enzymes which are responsible for the formation of different activities. It has a major role to play in the different inflammatory pathologies. Inflammatory pathologies of resulting in insulin resistance, adipokine actions, and it can also be, some of those factors could be responsible for the vascular disorders and particularly the vessel modeling, that reconstruction of the vessels in the healing process, and the insulin resistance, which can, or insulin sensitivity. It has a direct relationship with the mechanism of glucose metabolism and insulin resistance also is is controlled by, rather modified by, the adipose tissue, and hence adipose tissue has to be considered into two components. The one component, it adds to the weight and then accumulated, acts as energy storage. The other component, which is metabolically active. So when we say medo dhatu and mansa dhatu, the medo dhatu is that portion of the adipose tissue which is the storage function, and the mansa dhatu otherwise is the structures which are responsible for the other. Now of course we will have more discussion on this issue when we discuss further into the dhatus later on. So it's just a prelude. So medo dhatu, again I would not translate medo dhatu as adipose tissue, or I would not translate medo dhatu as the muscular tissue also, the other tissue. Rather I would translate the other way. Medo dhatu I would consider as the fluid and electrolyte mechanism as well as the adipose tissue, the two components. The adipose tissue component is the one which is responsible for the diseases like Saulia, whereas the fluid and electrolyte mechanism and that metabolic mechanisms, all that metabolic mechanisms of the adipose tissue, they are responsible for the prameha. That's how it has to be considered. So Meduja Vyadhis, when we discuss about that part we will have that kind of a discussion later on, and a part of that we had already discussed in the previous sessions. So I will not waste more time on that. But when it comes to the question of Mansadhara Kala, the issue of Mansadhara Kala, The Mansa word that suggests about the adipose tissue which covers the area of the body. That's the point. Then the next is the Raktadharakala. Raktadharakala is a मांसस्य अभ्यन्तरतः तस्यां शोणितं विशेषतस्य शिरासु प्लीह्नोश्च भवति वृक्षात् यथा अभिप्रहतात् क्षीरम् आवहेत् मांसादेवं क्षतात् क्षिप्रं शोणितं संरच्यते. Now within that soft tissues of the body, the vessels are located, and the mucosa of the vessels, the endothelial structures of the vessels, endothelial surface of the vessels, they would have their different functions of maintenance of the blood supply, and these are considered as a Raktadhara. Now, when you have the gross big vessels, they are seen directly. But when it comes to the question of capillaries, the capillaries may not be visible, and you will be knowing that there is a capillary or bleeding only when you cut the tissue, if you do not use a microscope. Of course, if you use a microscope, you can see the capillaries too, but if you do not use a microscope, you will be able to know the presence. You will be able to know. You will be able to know the presence of the blood supply only when there is a cut injury. Now that's exactly what Sushruta says. Vrikshadhyata abhiprahatat. As you cut the wood, you will be able to see the latex. Similarly, with the tissues of the body also, when you cut, you will be able to see the bleeding, and that's how it is confirmed. Like Mamsa Devam Kshatat Shipram Shonitam Saprachite. Because of that injury, you will be able to see the presence of the blood supply, and hence the blood vessels and the layers of the blood vessels, they have to be identified as a separate structure, and this has a relevance. This description has a relevance because the description is presented in a situation where microscopes are not available. Now, as far as the present day is concerned, we know the blood vessels have a very unique lining of membranes called as endothelial membranes, and these endothelial membranes, they are mainly of three categories: continuous, fenestrated, and sinusoidal. Mainly the sinusoidals in the capillaries, where there will be opening, and due to that presence of the opening, the contents of the blood will be flowing from the capillaries into the tissues, as well as the contents can flow back. Depending upon the size of the pores, there will be these pores as such. Then either it could be a smooth this basement membrane, or it could be a fenestrated membranes. Like as in case of the veins, the surface will be fenestrated. But the most important characters, the functions of these vessels, these endothelial vessels, will be, it will be prevention of the blood clotting. The endothelial surface have a property of avoiding the blood to get clotted, because the surface is extremely smooth. The endothelial surface provides a extremely smooth surface, as well as it gives a positive charge, so that the cellular elements are pushed towards the core of the vessel, and only the fluid comes towards the close, and this is what we call as the lamellar flow. The blood flow is maintained in a lamellar fashion, and maintenance of that will be mainly by the endothelial surfaces. As well as the endothelial surfaces also can prevent the clotting by adding some chemicals like heparin. Heparin sulfate is also a cofactor in the formation of the thrombin, and hence it keeps the blood non-clotted. Whenever there is a need for clotting, then the prothrombin is released from the surface, and hence it also has a factor to a role to play with the formation of the clot whenever there is a need. Or whenever there is a pathological entity, a disease condition, then there could be abnormal formation of the prothrombin into thrombin, and hence the clot could be formed. Inflammation also has a role to play with the endothelial surface. Endothelial surface are cells which give a signal to the white blood cells, so that they can come and produce a phagocytosis action. For inflammation, you need to have a phagocytotic action, and that phagocytotic action is triggered by the endothelial cells. Whenever there is an injury and then there is a healing, a new blood vessels have to be formed, and that formation of the new blood vessels also is a function of the endothelial cells. And of course, it also helps in the regulating the blood flow by constriction as well as the dilatation. Though the muscular layers have a major role to play, the endothelial surface also has a major role to play in maintenance of the peripheral resistance. So these are the main functions of the Raktadhara kala. Then Tritiya Medodhara Nama, Medo hi Sarvabhutanam Udarastham Anvastishtya Mahasutya Madhya Bhavati Stulatishu Visheshena Madhyato Abhyantarashritah अथ इतरेषु सर्वेषु सरक्तं मेदोच्यते शुद्धमांसस्य यस्नेह सा वसा परिकीर्तिता. Now, word medo again, we have already referred to that issue, mamsa and medas earlier when we discussed about the mansadhara kala. Medas word has both the meanings. The one part of the medas is that storage adipose tissues which are located in the abdominal wall and so on. But the other major important role of the medas has to play with the maintenance of the homeostasis of the body, and this is located in the madhya, the bone marrow. The medodhara kala, the major portion of that is in the bone marrow, and the function depending upon the stage and depending upon the location, the medas in the bone marrow may be as either reddish red marrow or it could be yellow marrow. Sthulastishu Visheshena Madhyapo Abhyantarashritah. In the sthulastic, major bones, long bones, it is presented in the interior area. Atha itareshu sarveshu saraktam medautyate. In the other areas, the flat bones, it is red in color, red marrow is seen, and that hence it is called as saraktam meda. Then Shuddha Mamsa Yasneha Savasa Parikirtita. Whereas the adipose tissue, in the adipose tissue, if you separate that, squeeze out the liquid portion or the oily portion, that's called as vasa. So these are the different byproducts of the medo dhatu. But more important about the medo dhatu is about the homeostatic mechanism, the mechanism of maintenance of the whole body's functions, as in case of the bone marrow. Of course, the other part is the adipose tissue, and the adipose tissue, vasa is the byproduct, whereas with the bone marrow cells, it's about the maintenance of the functions. Now, maintenance of the functions, when I say, usually we will consider that bone marrow functions as the hemogeneration or production of the RBCs. But it's not only the production of the RBCs. Production of the RBCs as well as the marrow cells, white cells, is a function of the bone marrow. That's absolutely true. But beyond that, there is another important function, important issue. The bone marrow has the stem cells, the adult stem cells. Stem cells are pluripotent cells, cells which can generate it and multiply and form different portions or different types of the tissue. The embryo itself is a basic stem cell which can produce any sort of cell, and hence it's called as a totipotent. But later on, as it divides, the cells become pluripotent, and among them we call them as stem cells. Now, the embryonic stem cells are the most important issue which are seen in the blastocyst stage, and they can produce any form of the tissues. In the adult body also you will have the stem cells, and the stem cells are located in many areas of the body. It's not only the bone marrow, but major amount of this adult stem cells are stored in the bone marrow. In addition to that, the stem cells also are present in the brain, blood, liver, and so on, skeletal muscles also. But the most important is of the adult stem cells, and the adult stem cells now are being used as a option of the treatment for many diseases even. And they are mainly pluripotent, they are located in the bone marrow, and that's one of the important functions. And these stem cells now are being used. This is one of the ultra modern form of the treatment aspects, interventional aspects, is stem cell transplantation, and it is now used in maybe is supposed to be a treatment, choice of treatment in the future situations for large number of conditions like stroke, brain diseases, degenerative diseases of the brain, teeth degeneration, and wound healing, bone marrow transplantation. Of course, it's established now. Then spinal cord injuries, muscular dystrophies, diabetes also may be treated, so on. So that's the future area, and that's exactly what Sushra has considered as the medo dhatu as such. So it's not that medo dhatu is only the adipose tissue. Adipose tissue is a small part of the medo dhatu, But whenever it comes to the question of Medo Dhatu as a whole, we have to consider the broader vision of the Medo Dhatu. It's not only about the adipose tissue, the other functional aspects also were considered. More of that discussion when we go into the Medojanya Vyadhis like Prameha. Again, I think we have already discussed that in the Prameha Nidana. That's the point. Next is the Shleshmadhara. चतुर्थी श्लेष्मधरा नाम सर्वसन्धिषु प्राणभृतां भवति स्नेहात्यक्ते यथा ह्यक्षे चक्रं साधु प्रवर्तते सन्देहः साधु वर्तन्ते संश्लिष्टाः श्लेष्मणा तथा. Now this again is a very unique structure in these joints, particularly in the synovial joint. The structures of the synovial joint would be having that ligaments which with the mechanical support, but the interior there is a membrane, the synovial membrane. That synovial membrane tissue looks like epithelial, but it's not typical epithelial. It's having multiple tissues like fibroblasts, macrophages, lymphocytes, T lymphocytes, and the also catachrophages, like very unique cells of the synovial membrane, which are the macrophages of the tissues there, and hence it's a rather complex layer. Often synovial membrane is often written as a epithelial membrane. It's not really epithelial. It has multiple structures, multiple cells together, and hence synovial membrane is a unique membrane where you will have multiple varieties of the cells present over there, and it's having a lining. It covers the lining of the joint formation, and it forms a synovial fluid which mainly contains hyaluronic acid, which can prevent the coagulation there. The functions of the synovial membrane are mainly to keep the joint functioning smoothly and as part of the lubrication agent, as well as, but more important than it can prevent the inflammation. In diseases like inflammatory pathologies, the abnormality of the synovial membrane is the first sign of many of the diseases of the joint. And before the joint affects the bones, synovial membrane involves. And if you can identify these in the early stages of the degenerative pathologies as well as the inflammatory pathologies of the joint, particularly inflammatory pathologies of the joint like rheumatoid arthritis, etc., the first of the structure which gets involved would be the synovial membrane, which will have either lymphocytic infiltration or villus formation or a revascularization, neovascularization. These three changes are seen in the early stages, and hence they can be of some use in making the diagnosis. Next is the Purishadhara Rama. Panchami Purishadhara Rama. Yo antakosthe malam abhivyajate pakvashayastata yat samantat kostam tada antrani samashidaha undukastham vibhajate malam maladhara kala. Purishadhara kala is the membrane which helps in maintenance of the fecal contents. Now, at one stage, Sushil says it's about the presence in the Purishadhara, the area of the formation of the feces. But at the same time, the kala, which helps in maintenance of these functions, is seen Yakram Samanta throughout the costa. The layers also may be named as Purishadhara, though by literally meaning they are not related to the feces directly, but indirectly the formation of the feces starts from the moment you consume the food. The food is gradually converted into the feces. In between you have that long process of the digestion and absorption. And based upon that digestion and the absorption, the structures of the membrane, the mucous membrane, would be varying. In the stomach, you will have the different gastric glands which will add the secretion of the acids. The pitta is added. Later on, it results in the absorptive area. In the intestines, you have the villus structures. In the colon, water absorption is a major area, and depending upon that function, the modification of the cell, these mucous membranes are seen, and these mucous membrane modifications can be seen under the this microscope. So Sushruta has made that idea of a generalized description, yakrat samantat kostamcha. Tata antrani samashitaha, starting from the yakrat to the intestines. Maybe we may add up from the mouth to the these intestines, large intestine. They are the Purishadhara, but very specifically Undukastham, when it is in the Unduka, the distal portion of the intestines. Unduka is the globular portion of the intestine, maybe the cecum. Now, of course, now it is translated as cecum. Now, so it's not only the cecum, beyond the cecum also, the large intestine portions, which where they really are having the feces, the ultimate products of the absorption being complete, the residual parts of the food. That's Maladhara Kala. So, maybe Maladhara Kala, Purishadhara Kala, may be considered as the mucosa of the colon or mucosa of the whole of the gastrointestinal tract. Both of them are justified, and based upon the typical functions there, the structures are modified and the structures are presented over. Now again, Pittadhara Kala, the Shashti Pittadhara Kala, another of that, very specific. Social has considered this as a the Chatvidam Annapanam Amashaya Pratyutam Pakvashaya Upasthitam Dharayati. From the Pakwash, Amashaya to the Pakvashaya. There again you have a specificity. The specificity starting from the Amashaya to the Pakvashaya, the intestinal absorptive area, that's called as the Pittadhara Kala. Ashidam Khadidam Pitam Leadam Kosagadam Radam Tajiri Yatha Shoshidam Pittadesa. Absorptive functions. Now, I think I am not producing the confusions. The issue is when it is the Maladhara Kala, it is about the excretory function, where the residue is pushed off. When it is the Pittadhara Kala, it is about the function of the digestion and the absorption. Digestion and the absorption are depending upon the type of the food, consistency of the food. The process of the digestion would be varying. When you eat a hard substance, that has to be mixed and homogeneous. So you will have to have a gurgling movement, and that has to be broken down, and ultimately it has to be produced into a homogeneous fluid. It can be absorbed, and hence the justification of that classification of the food into Ashita, Khadita, Pita, Lida, something which can be drunk, liquid, or something which is having a thick consistency, which has to be chewed, and then it has to be bitten, and so on. So that's the one part of it. But from our point of view, discussion about the Kala, the Pittadhara Kala suggests about the digestive and absorptive structures, mainly the villi, lacteal, and so on, where the substances are absorbed. I think I will not go into the physiology, basic physiology of that, probably you know about it. Then the Maladhara Kala is when the residual substances are pushed out, or if you have to be more rigid, Maladhara Kala can be attributed only to the large intestinal planes. The small intestinal mucosa could be considered as a Pittadhara Kala. I will not go beyond that for the argument. The argument, both the ways it is justified. Maybe it's better to accept the version of duality. Like when it comes to the question of excretion, it is the Maladhara Kala. When it is the question of absorption, it is the Pittadhara Kala. Then Saptami Shukradhara, ya Sarva Praninam Sarva Sharira Vyapini, Yatha Payas Sarpistu Gudhascha Ikshavaso Yata Sharireshu Tata Shukram Dranam Vidyabhishapuraha Jangule Dakshine Parshe Bastidvarasya Japyadaha Mutrasrota Yatha Shukram Purushasya Pravartare Krishna Dehashram Shukram Tasarna Manasastada Strishu Vyaya Chadaschapi Harshat Tat Sampravartare. Now this is again issue which we have discussed in the previous session about the semen and the spermatozoa, or the production of or capability of reproduction. The capability of reproduction is a hormonal issue, and it has a very generalized effect, starting from maybe the stimulus from the brain and so on. It's a complex issue. Now from Ayurvedic point of view, the process of ejaculation of the sperm starts with that kind of a complex mechanism, neurohumoral mechanism, where the starts with the sensations from through the nerve endings, then the hormonal changes, and then there is an ejaculation of the sperm which is formed at a different structure. So formation of the sperm and the storage is one part, and the capability of reproduction is another part. Shukra Dhatu is about the other that component of neurohumoral mechanism which is present throughout the body probably, and the Shukradhara Kala, the membrane which is seen, is about the genital area, and these structures, the membranes, would be again modified depending upon the functions. Like in the testis, you have the Sertoli cells which can divide and then form these haploid sperms, and the sperms have a function of moving through the spermatic cord, and the structure of the mucosa of the spermatic cord is modified in such a way that they can facilitate the movement, and the seminal vesicles, the sperm has to get matured, and there will be addition of those substances, different chemicals which are added to facilitate the environment of the sperms for the reproduction, and hence based upon that the structures of the mucosa, the color, Shukradhara color, can vary from testis to seminal vesicle based upon the functions, and all of these can be considered as a Shukradhara color. Whereas Shukradhatu is the capability of reproduction, which is a complex neurohormonal mechanism. With this, I think we conclude today. The next part is about the other aspects of the Garbha and the Garbhadharana. If there are any questions, I'll try to answer, and then we'll conclude today. There are many questions. In the Tvaka, thickness said by Sushruta varies according to region, and which pramana is Vrihi pramana is about the abdomen. That's what Sushruta already has clearly. That I think we have referred to that sentence. In the Udara, when it is in the context of drainage paracentesis, Vedana of the Jalodhara, Sushruta refers to that issue. You will have to penetrate up to a level of Vrihi. So Vrihi pramana is referred to the abdominal area. That's very clear cut. There is no need of confusion. Now, whether really it is the same in all patients or not, that's not true. Of course, there you may have argument on that. It can be thicker in some, in maybe thinner in some, but it's an average. When Sushruta mentioned about the Vrihi, it's the average thickness of the abdominal wall. Then the next is those stem cells are present in brain. When the brain cells death occurs, why there will be no regeneration? Of course, that regeneration of the nerve cells, it's now a area where there is a lot of new development. Earlier it was believed that the nerve cells do not regenerate, but the recent researchers have shown that nerve cells also can regenerate, but regeneration is a very complex process there, and it requires a very specific stimulus. Regeneration of the nerves now is a fact which is proved. So I think whenever there is a opportunity, we will refer to that. There is new researchers in that area. Lots of researchers are already seen, and many reports have come up where regeneration of the nerve cells is possible, but the environment for the regeneration and the stimulus for the regeneration is not the same as that of the other tissues. It's a very complex process. So regeneration is possible, and the role of the stem cells in the regeneration also is now proved. Earlier, a few years back, of course, this was the theory where the nerve cells would not regenerate. Then could you please light on importance of stem cell preservation from umbilical? Okay, that umbilical cord preservation is, of course, that issue is a issue which is not related to what we discussed, but anyway, because you have asked, like in brief, the whole issue is a very complex issue. The preservation of the cells, umbilical cord stem cells, is to maybe in future to generate the tissue organs of the different bodies. So wherever there is a tissue loss, in general, in brief, now of course, many times that kind of statement may have many exceptions, but in general, whenever there is a tissue loss and you want to have a regeneration of the tissues, embryonic stem cells are used, and the embryonic stem cells are preserved. So there is a very standard technique of maintenance of this preservation of the umbilical cord collection, as well as maintenance of these stem cells of the umbilical cord, which can be used. So that's in brief. More details about that whole protocol maybe we may discuss on some other day. Then the other is, what could be the reason for mention of Pittadhara and after Purishadhara? One after the other, I don't know. So it cannot be just simultaneously. Maybe you, from our current point of view, we may consider the logic like the digestion and absorption occurs first, and the feces would get separated, and you feel that it's more rational to consider Pittadhara Kala in the beginning and Purishadhara Kala in the next. But if you consider the other way, the formation of the feces as the most important part, the absorption as the next important part, then it's justified. So it's the way you look at it, and I don't give any significance to what's mentioned beginning, what's mentioned in the next. That's all the point. It's important that you have to mention all the details and it's mentioned there. I think that we need not create any controversy on that part, like right. uh, that's about it. So we'll conclude today. Thank you.